Okay, so welcome to the presentation. Here we'll be discussing on the two types of genetic drifts. Okay, we have been discussing about the evolution, isn't it? And we saw that the most important factor that influences evolution or that results in evolution is the natural selection. But natural selection is always, to some extent, it is biased in the sense that natural selection tries to favor the favorable characters or the useful traits or present in a population. And usually those individuals without those traits or with what you call unfavorable variations of a particular trait are removed from the population, right? But there is yet another process that influences evolution, and that is genetic drift. But in genetic drift, what happens is we never can be sure that this particular trait or a favorable trait is going to get selected, or individuals with a favorable variation of a trait is going to get selected. It is purely random process. That is, genetic drift is a random process wherein every genotype or every variation of a particular trait can get selected or can get completely removed from the population. Okay, and there are two main kinds of genetic drift. One is bottleneck effect and the other is founder effect. Okay, bottleneck effect, as here we can see, here there is, just imagine, this is just a representation where each of these colored beads, it represents the variance in the population. Okay, so here we have two variants in the population, right? Blue colored and yellow colored beads represent two variants of a particular trait in the population. Okay, so this is the parent population. Now what happens is, maybe because of a disaster, like maybe like a flood, or it may be because of like any natural disasters or any accidents for that matter. Just imagine, because there is the drastic reduction in the population and only very few individuals survive, while the rest of the population, major part, it gets completely destroyed. Okay, you can see that here almost equal number of yellow and blue colored beads are there. But what happens when there is a survival? What happens is, it is more of blue colored, only two yellow colored beads are there, isn't it? So this represents surviving individuals, where the individuals with a particular variation is more as compared with its complementary ones. I hope it is clear, right? Now it is so that you can see that only very few are surviving, and among the surviving ones, a particular genotype is more predominant than the other one, while the rest of the population, major part of the population, is destroyed in the catastrophe. Okay, and from these surviving individuals, the population begins once again. Okay, it increases back to the normal population number. But what happens here is, you can see that since the surviving individuals have like more individuals with a particular variation, obviously they will reproduce and pass on this particular variation to the generations, isn't it? So more number of individuals with a particular genotype can be found in the coming generations. Clear? So here we have 50% of the yellow and blue genotypes, isn't it? But by coming here, the new generation will have more of blue genotype than the yellow ones. Okay, this is known as the bottleneck effect, because it is somewhat like beads filled up in a bottle. When it is tilted, obviously very few gets escapes out, and we can't be sure that it is equal number of yellow and blue beads going to come out of the bottleneck, isn't it? So it could be it is somewhat like permutation combinations, isn't it? So this is what is known as a bottleneck effect. Here a very particular example is with regard to northern elephant seals. So this is a seal here. The northern elephant seals have reduced genetic variation, probably because of a population bottleneck humans inflicted on them in 1890s. So in 1890s, humans had been taking up hunting of these seals, and these hunting reduced the population size to as few as 20 individuals at the end of the 19th century, and the present day population of about 30,000 has come up or has originated from these 20 individuals. So whatever genotypic predominance had been shown in this particular 20 individuals had passed on through the generations after generation. So, their genes still carry the marks of this bottleneck, and they have much less genetic variation than a population of southern elephant seals that was not so intensely hunted. So, before 1890 what was the population, and after 1890 or in the 19th century what is the population? If we compare, obviously the genetic variation is far less in the present day population as compared with the what you call the previous populations. Okay. Here you can see in the figure that how many it is like 1, 2, 3, 4, 5, 6, 7, 8 and 9. So, out of 9 only 2 are yellow, isn't it? If we take it is almost 20%, almost 20% is the one with yellow colored genotype, isn't it? While blue colored genotype, it is almost 80%, right? So, 80% of the population, here it was 50%, 50%, but it came down to 20 and 80, or it got shifted to 20 and 80 with predominant of the blue genotype. I hope this is clear to you. So, it is a genetic drift, isn't it? Now, yet another what you call a type of genetic drift is the founder effect, and here it is not because of any what you call the catastrophe, accidents or disasters, but it is because of migration. Okay. When a few individuals or a small group from a main population migrate to a new area, only a limited portion of the parental gene pool is carried away. Okay, that is yeah. So, a small number of individuals migrating from a larger population, this smaller population, it leaves the main populations and moves away to a new area, okay, and it starts to get established over there. A smaller number of individuals which had migrated may not have the same genetic frequency or genotype frequency as that of the larger population. Okay. If the larger population is having for a particular genotype 50% of dominant and 50% of recessive, or 50% dominant allele frequency and 50% recessive allele frequency in the main population, the smaller population which migrates out needn't have the same kind of frequency. Okay. It may be like 10% of recessive allele frequency and 90% of dominant frequency, or it may be the other way around. Okay. So, in both cases what happens is the migrated population may start to reproduce, and it may give rise to a larger population in a completely isolated area. There the genetic frequency or the allelic frequency of the new population may not be the same as that of the parent population. Okay. That is actually what we refer here as the founder effect. Okay. So, founder effect means the allele frequency or the genotypic frequency of the founder of the newer population. Who are the founders here? They are the migrants from the older population are the founders. Okay. So, the what you call the genotype frequency of the founders will affect or will be reflected in the genotype of the population which is going to develop from the smaller population. This particular concept was introduced by Ernst Mayr in 1956. So, in a small migrant group, some genes may be absent or occur in such low frequency that they may even be lost. The unique frequency of genes that arise in a population derived from small bands of colonizers, or what we call as founders, has been called the founder effect, or it is otherwise referred as founder principle. Okay. This principle was proposed in 1956 by Ernst Mayr, who was the Harvard evolutionist, and the founder principle essentially emphasizes the conditions or circumstances that support the operation of genetic drift or Sewall-Wright's effect. Okay. There are plenty of examples here. We can one example is like given over here. It is with regard to the Afrikaner population. Afrikaner population of Dutch settlers in South Africa is descended mainly from a few colonists. Okay. Today, the Afrikaner population has an unusually high frequency of Huntington disease, or gene that causes Huntington's disease, because the Dutch colonists who happen to carry that gene with the unusually high frequency, and this effect is easy to recognize in genetic diseases, but of course, the frequencies of all sorts of genes are affected by founder events. Another example is about the North American Indian tribes. North American Indian tribes for the most part lack the gene that governs B blood group. Okay. However, in Asia, which is the ancestral home of the American Indians, the B blood group gene is widespread. So, the ancestral population of Mongoloids that migrated across the Bering Strait to North America might have been very small. Accordingly, the possibility exists that none of the prehistoric immigrants happened to be blood group B, and it is also likely that a few individuals of the migrant band did carry the B gene, but they failed to leave descendants. Okay, so that is yet another observation on the founder principle. So, you can find that the founder principle or the founder effect and the bottleneck effect are two major kinds of what you call genetic drift, but it is because of two different reasons. In bottleneck effect, the subpopulation is caused when a natural disaster reduces the size of an original larger population. Okay, that is original larger population, only some of the individuals survive, the rest are completely removed. Okay, and this may be because of habitat fragmentation or over exploitation of species or any other kind of disasters. But what happens in founder effect? In founder effect, the founder establishes a subpopulation as a new colony drawn from a larger population. Okay, so here what happens is the larger population is remaining, but a few individuals from the larger population moves out. Okay, so here the individuals which moves out and which are known as the founders and the parent population both are surviving. Okay, no one is getting removed from the removed. Okay, so here both are remaining and founder, the smaller population which moves out, the subpopulation, they form the founders. Okay, it is the resultant product of migration of species. Okay, so this is the different types of genetic drift. Fine, thank you.